Okay, let me start recording. Okay. Okay. So let me first start with the lack operon, right? First, G video. Could you send the guidelines for the G video presentation? Yeah, definitely. Yeah, definitely. Those information about the GE scientific communication assignment. The more details I will form it and try to send to you. Just now I discussed with just rough idea, and then clear details I will let you know. More details I will post it. Okay, so the first one is lack operon. So lack operon is having a common promoter, and it's having like Z, I think Y and A, something like this. And these are the three genes which are regulated by a common promoter and a common terminator. Okay, so this is how it was. It is present in the bacteria, so that whenever the any gene of lacoperon is required, all three will be transcribed, all three will be translated, and all three uh, will be utilized in the lac. Okay, lacoperon expression whenever it is required, and it will be utilized in the lactose catabolism. So these genes are required, these proteins are required. So after the transcription is like this, and it is going to be translated is another protein, this is another protein. So this one is understood that it will break the lactose into glucose plus galactose, and this one is a permease. It is allows lactose to enter the cell, and this is an unknown function. This is not known. Okay, whatever it is. So these three genes are present as a common promoter. So these all, see, these two at least is required when the lactose needed for the cell as a carbohydrate source, then only it is required, isn't it? So that is why it need to be switched on all the time, not all the time, at a time whenever it is required, or switched off whenever it is not required. So that is how it bacteria try to regulate the all the genes at a time. And there is another gene separately from this operon, that is LAC-I, which is producing a small mRNA and which is producing a small protein, and we symbolize something like this. Um, this protein is called as repressor, and this repressor, few repressors together, four repressors together, it will form a active repressor, it's a tetramer of repressor is the active repressor. Okay, tetramer of the repressor is the active repressor. Okay, what happens? This one, always keep expressing, always keep binding to this location, so that RNA polymerase cannot bind to the, this RNA polymerase cannot bind to the promoter region. When there is no, see, this is always in the sense when glucose present. So, okay, nothing to do with the glucose when lactose absent. So when lactose absent, instead of always, then we can say lactose absent. Whenever lactose absent, what happens? Keep on the small mRNA, like I keep um, expressing and keep expressing the repressor protein as a tetramer of repressor, which is act as a active repressor and goes and block to the promoter region. And usually we call as a small region of the promoter is the operator region of the promoter. It occupies. So RNA polymerase is not going to bind, and it's so there will be no transcription of lac operon. So when there is no transcription of lac operon, what happens? There will be no this beta galactosidase and permease and etc. All these things enzymes will be not expressed. So there will be no utilization of the lactose for the energy source. When there is no utilization of lactose as energy source, when there is no absent, there is absent of lactose. It's sensible, right? When the lactose is not present, so no need to use lactose as an energy source, and no need to have enzymes to use lactose as an energy source. So that is what happens. And when lactose is present, so when lactose is present, so when the lactose is present, the lac I is not no change, still will be expressing irrespective of any condition. Lac I will be keep on expressing the repressor molecules and produce this one. But when the lactose is present, then what happens in the environment? So bacteria is there. So if the environment lactose is present, so this will give this a little bit leaky entering of the cell, and the lactose will be present inside the cell a little amount. Those lactose molecules, when the lactose is present, lactose will convert into allolactose. This is the isomer of it. This allolactose having the affinity with the repressor molecule or the allosteric site. What is the allosteric site? The other site of the protein where it is having the binding site. So this is the binding site and the other site. Okay. So it is having the affinity to the allosteric site of the repressor molecules. Once it binds, it changes the conformation such a way it will not be able to attach to the just to make you to imagine only this one. Okay, it's not the exact structure of it. Okay, so allolactose, when it will be present, when the lactose is present in the environment. So when the lactose is present, so some of the lactose will convert into allolactose, which is an isomer of it, which is having the affinity with the allosteric site of the repressor molecule. So it will bind to the repressor molecule, and it will change the conformation such a way that it will no more attach to the promoter region of the operator region of the promoter of the lac operon. So then this will be going to detach it. Okay. So once it gets detached, what happens? So RNA polymerase can able to bind and transcribe. So there will be transcription of the lac operon. So when will be the transcription of the lac operon? When the lactose is present. So that is about the the promoter blocking and unblocking, and presence of lactose and absence of lactose. So there is another issue. This RNA polymerase, even though the promoter is free, the RNA polymerase alone cannot bind to this region. This region having the affinity such a way that RNA polymerase alone is not going to bind to the promoter region. So whenever it binds, it fall off. It's a loose affinity. Affinity is not that strong to bind to the region, so it fall off. So what it required? It required some other molecule to bind to this promoter, this particular promoter. When I'm talking about the affinity of the promoter to the RNA polymerase, only this promoter, not other promoters of the bacteria. So that bacterial, that other genes promoters may be strong enough to hold the RNA polymerase, same RNA polymerase. But this lac operon promoter is not strong enough, the affinity, to bind to the RNA polymerase strongly. So it required some other molecule to be hold, to attach to the promoter region. So that molecule is, so you can put anywhere, just, I mean, exactly not here, not here, anywhere. So it required some conformational change in the thing so that it will attach strongly. Though this molecule is called as activator. Okay, so what we call this allolactose molecule as, because allolactose molecules are going to bind to the repressor molecule to detach it. So it is detaching, mean it is again initiating, so inducing. So that's why allolactose we call as inducer. Okay, so allolactose called as inducer, and this kind of regulation is called as inducible negative regulation. Negative regulation, when we call, when the repressor is blocking the promoter and unblocking, this is called as negative regulation. And when the inducer is playing a role in this negative regulation, then we call as inducible negative regulation. So when the lactose is absent or present, we can say inducible negative regulation, we can say. And that is about it. And this one, we need an activator molecule to strongly bind, even though the promoter is free. Even though the promoter is free. Okay. So irrespective of the lactose presence or absent, activator molecule is compulsory for the initiation of the transcription. That is what the underline. So that is what the message we need to understand. So what is this activator? So this activator is kind of a cyclic AMP. This one will be only synthesized in the cell in absence of glucose. Activator molecule will be produced in the cell in absence of glucose. So now the idea is glucose presence and glucose absent. So when glucose is present, Okay, when glucose present, what happens? No activator. When there is no activator, what happens? This RNA polymerase fall off from the region, and there is no transcription. Though promoter is free, but there is no activator molecule, and there will be no transcription of operon in presence of glucose. And when glucose absent, activator molecule will be synthesized, and activator molecule along with the RNA polymerase will bind to the promoter region strongly. Thus, it will initiate the transcription of lac operon and go for the synthesizing the proteins which are needed for the catabolism of the promoter utilization of the lactose as an energy source. So that is what you need to understand. Presence or after absence of lactose and glucose, presence or and absence of glucose. So we'll try to understand in a summary what happens. There are four possibilities, right? So I'm erasing here. We are having four possibilities. Glucose present, lactose absent. Glucose present, lactose present. Glucose absent, lactose absent. Glucose absent, lactose present. So these are the four situations you come across, the bacteria come across. What happens in each situation? So when glucose is present, there will be activator molecule. So that means it is having the RNA polymerase with activator. It is there, so that it will strongly bind to the promoter region. But what happens? Lactose is absent. When the lactose is absent, what happens? The repressor molecules are going to bind to the promoter or operator region of the promoter and blocks. So even though the RNA polymerase and activator is ready to strongly bind, but promoter region is not free. So that's what happens. There will be no transcription. Or no, there is no transcription of lac operon. So lac operon, no transcription. What happens in the so glucose is present, again the same thing, it will be having the glucose present, sorry, glucose present, there is no activator. Sorry,
yes, that's why I was wonder.
Yeah, sorry, I just sorry. Okay, so there is no activator. Of course, whatever it is, the result will be the same, no transcription. But in understanding, so when the glucose is present, no activator. So no activator, but promoter region also blocked, both. So lactose absent, because of that operator region is fully blocked. At the same time, RNA polymerase also cannot bind to the promoter because there is no supporting molecule. The same thing happens to the here. So glucose is present. That means RNA polymerase alone is present and lactose present. So when lactose present, what happens? So no block of promoter with repressor. So repressor molecule will be detached. So because inducer molecules are there. But the problem is RNA polymerase alone cannot bind and transcribe because there is no activator molecule because glucose is present. So it will result to no transcription. And the third situation, glucose absent. Yeah, here is the one we have RNA polymerase and activator molecule is there. So here it can bind, it can able to bind to promoter. Okay, but the problem is lactose is absent. When the lactose is absent, same thing happens, bind to the promoter and blocks. So blocking of the promoter region. So though the activator molecule is there, it is not free to bind to the promoter. So it will result to the same no transcription. And yeah, glucose is absent. That means it is having the RNA polymerase and also activator molecule. And lactose present, that means allolactose is present, and which is acting as an inducer molecule, and there will be no blocking of operator of promoter. So then this is yes transcription. So that is the summary of the lac operon regulation, how the bacteria senses the signals whether the lactose present or absent, or glucose present or absent, and based on the situation, four possibilities: when the lactose present and glucose absent, only the condition how bacteria can able to transcribe and translate the lac operon genes and utilize the lactose for the energy source. Otherwise, what happens in four conditions, it's keep on expressing, the other three condition is not expressing. If it's four condition keep on expressing, there's a lot of energy, right? There's a lot of amount of nucleotides need to be utilized for the uh, RNA synthesis, mRNA synthesis, and protein synthesis, and unnecessarily when it is not required. So this is one of the excellent example how the bacteria utilize the energy efficiently whenever it is required. So that is one of the nice example for the gene regulation, simple gene regulation in bacteria. So that we can see in almost all living organisms, but in bacteria we can see in operant kind of way, and in eukaryotic system in much more complex mechanisms are there. Maybe I mean, yeah, if time permits in one of the course, we can try to discuss about it. Yeah, so this is about lac operon, and we'll try to talk about tryptophan operon. Okay, let me see. Okay, tryptophan operon. Uh, tryptophan operon is also at a two-stage uh, regulation. One at the promoter level, if that miss any case at the transcription level regulation. The two kinds of regulations are there. So let us first understand how the tryptophan operon genes are present. So this is the promoter. and there is some region which is not coding for any genes needed for the tryptophan operon, and followed by 5 genes, 1, 2, 3, 4, 5. So we call as E, D, C, B, A. So these 5 genes are needed to synthesize 5 proteins, 5 enzymes which are needed for the synthesis of tryptophan. So try to understand that here is an anabolic reaction where tryptophan is synthesizing. In the lac operon, lactose is utilizing for the energy source. Here, giving the energy, utilizing the energy, uh, making the complex molecule for the simple molecules, uh, that is tryptophan, that is called as anabolic reaction. Okay, so yeah, there should be some promoter. This should be an RNA polymerase. Okay, so which will going to bind and the promoter and transcribing and translating the mRNA. So mRNA is going to be a single mRNA from here to here, from here until the end. So once the RNA binds here, it will start transcribing from here and keep on transcribing until the end. There will be some terminator signal to stop it. And after transcribing everything, so this region will be some having some ORF, this another ORF, this another ORF. So this will be translated by the ribosomes into different proteins. Okay. So these proteins are needed for the synthesis of the tryptophan molecule. So what you understand now, these proteins are needed when the tryptophan is needed to be synthesized. So tryptophan biosynthesis, when this operon is transcribing and translating, then it's going to be biosynthesis of the tryptophan. So here also one regulatory repressor molecule is there, so which is TRPI or E something. So not E, I, or not even I, something repressor molecule, R I think. Okay, whatever. So it is a small mRNA and which is producing a repressor molecule. So this repressor molecule, so just to make you, imagine and visualize. So it will be present as a dimer. This dimer is such a way, this conformation is not going to be exactly fit to the promoter region. This conformation is not enough to strongly bind to the promoter. So what happens? The RNA polymerase will bind to the promoter and transcribing, even though the repressor molecule is going to be transcribed parallelly. That is not enough to stop the transcription. What happens? Okay, this is the scenario. So the tryptophan is less, or in a starving condition of the tryptophan, or less tryptophan, just better, less concentration of tryptophan in the cell. So less or low. Okay. So then tryptophans are low. What happens? The repressor molecules are going to be synthesized and producing a dimer. This dimer cannot bind. RNA polymerase will bind to the promoter region, and going to be transcription. and translation, tryptophan biosynthesis, and producing a lot of tryptophan molecule. When a lot of tryptophan molecule keep producing, so at certain concentration, it's a huge number of tryptophan. I'm using the same symbol. Don't confuse. This is lactose. Okay, tryptophan molecules have been synthesized a lot, and the cell inside the cell, a lot of tryptophans are present. Then, when the tryptophans are high, then what happens? These molecules bind to the repressor molecule at allosteric sites. Then it will change its conformation such a way that it can able to bind to the promoter region. And when it blocks here, so RNA polymerase cannot bind to the promoter region, and it will be not having free uh, to, uh, to bind to the RNA polymerase. So thus, there will be no transcription and no translation. Of course, there is no transcription, there is no translation, and there will be no biosynthesis. So that is what actually wanted. When the tryptophan high, no tryptophan biosynthesis. How? Tryptophan molecule itself act as a co-repressor. This one is a repressor, and this is a co-repressor. This one is apo-repressor when there is not active. Though this is going to be active repressor molecule. So then it will lead to the no transcription of the tryptophan operon, then leads to the no translation of the tryptophan synthesizing enzymes. So there will be no synthesis of tryptophan. That is what. So that is one kind of regulation. The second regulation is, so though the promoter region is blocked, then already by the time RNA polymerase already started the transcription of the uh, the tryptophan operon, which is very long and a lot of energy need to be utilized for it. When already there are a lot of tryptophan molecules and bound to the apo-repressor to make it into active repressor and block the promoter region. By the time RNAs are already, some of the RNA polymerases bound and keep started transcription process. So this not no transcription or yes transcription, this transcription is still going on. So what happens when the transcription is still going on? When this one is here, so the one from here, RNA, the transcription starts and still is going on. So when the move it is here, and this still it will be keep going on. Okay. Then when it reaches somewhere here, RNA polymerase moves uh, until here. Okay. And it's moved, RNA already synthesized a small amount of small stretch of RNA only, not fully, which is very long. If you want to, though the just picture showing a small blocks of 5 genes, but it's in fact is longer than this one. So what happens if you want to keep going and synthesizing all the genes and which will be translating? It's consumed a lot of energy, though it is blocked. So then the cell choose another screening at the transcription level, so while it is keep going, the RNA is going to be folded such a way that it is having a secondary structure. Okay, here let's see this transcription keep going irrespective of whether promoter blocked or not blocked, right? Until here, the RNA polymerase can reach whether the promoter blocked or unblocked. That means whether tryptophan low or high. So now we will try to understand what happens at this position when the tryptophan is low and when tryptophan high. So just I'll try to erase this one and try to understand the situation when the tryptophan high and low. So both conditions we'll try to understand what happens to the extension of the transcription. Until here it will go. So after that what happens? So this RNA, as I mentioned to you, this will be not present as just linear RNA. It will not just present like a GU. So it will not just present as a linear one. It will be present as a secondary structure. So in the secondary structure was such a way when so this is called a secondary structure. What is this? So stem loop structures mean there will be some nucleotides which are having the pairing. So right, in DNA it is having A to T, in case of RNA A to U and G to C. Though these kind of stretch of nucleotides are present somewhere near to the same sequence, so it will try to have a pair, and that's how it will be more stable than linear. So this kind of structure will be formed when it is keep going over there. And here is an ORF, a small ORF is there. Open reading frame, and here the end, UGA, I think, UGA. Something like this is there. So when the when the transcription is still going on in bacteria, as I told you, in bacteria, transcription and translation, everything occurs at cytoplasm only. So there will be no waiting of the finishing of the transcription and the transport of the mRNA from nucleus to cytoplasm, which you can see in the eukaryotic system. So in eukaryote, all this transcription need to be finished. Then only it need to be enter into the cytoplasm to initiate the translation. But bacteria, it is not like that. Bacteria, when the transcription is still, the RNA polymerase somewhere here, still going on, the ribosomes start binding to this location and start translating the things. And this, as I told you, it's just a small peptide. The peptide is not required for the protein, but this translation is very important to understand the tryptophan molecules are present inside the cell or not to the cell. So that is why this peptide is useful for the bacteria. So RNA polymerase binds here and keep moving because this is the RBS site, ribosomal binding site. It will bind, and it will start from the translating AUG until this is a serine codon is there. After that, a stop codon, and RNA ribosomes fall off from here to here. So when there is no issue, of ribosomes need to be stalled, then there is no problem for the cell. Then that means when tryptophan high. Why we are talking about high? There are two codons which are needed to be added two amino acids, tryptophan amino acids to this peptide, tryptophan and tryptophan. So this is a small peptide, will be synthesized. So the ribosomes move from here and until here. When the ribosomes are moving without any problem, there will be no problem with the structure. So this structure will be not disturbed. Ribosomes will be going through and very fast it will reach here, and when moving here, only this structure temporarily disturbed, but it will be keep going and it will be attaching here back, only here some temporarily disturbed and temporarily disturbed, and it will be going on. So this temporary disturbance will not disturb the structure. So just ribosomes keep moving and translating into small peptide we call as leader peptide, and each contains two tryptophan amino acids, and ribosomes will fall off. Ribosomes subunits will fall off after translating into leader peptide when tryptophan is high. And when tryptophan is high, when there is no problem in the structure, and this is actually a row independent terminator. So what is row independent terminator? This RNA polymerase, when it is reaching somewhere here, and it is having the stem loop structure and poly U, RNA polymerase fall off. So here it's fall from here. So falls from here mean, what does it mean? There will be no transcription. Okay, that is what happens when tryptophan is high and the RNA polymerase. So result that the growing mRNA is folded such a way that it is having a row independent terminator at this position, and the ribosomes keep binding to the RBOS site and initiating the translation at AUG and translating at the position of the tryptophan, and until the end there is no issue, there is no change in the secondary structure of the growing mRNA. And when there is no change in the structure of the growing mRNA, this will act as a row independent terminator. RNA polymerase fall off. Once RNA polymerase fall off, there will be no transcription. No transcription means no synthesis of these enzymes which are needed for the tryptophan biosynthesis. That is what required when the tryptophan is high. So in other condition, what happens? In tryptophan is low. When the tryptophan is low, so that means, of course, it formed the secondary structure similarly like this, but the ribosomes bind here, again the same, and keep moving. And when it is reaches to here, either this position or this position, what happens? There will be no sufficient tryptophan tRNAs carrying and adding the tryptophan molecules to the leader to synthesize the leader peptide, and the ribosome stalls there, stucks over there. So when they stuck over there, see, this structure is going to be disturbed. You can understand this. The pairing of the nucleotide is not going to be how the hydrogen bonds with the other nucleotides in the loop structure. So it is not going to be happen there. So with that, so it will RNA need to find the other alternative stable structure. So how it will find? So how it forms, it is going to be formed in different way. So just something like this. Okay. So what is here happening? So ribosomes are going to be stalled somewhere here. So, and it's having a different stem loop structure, polyuse here. So I was, as I was telling multiple times, so row independent terminator is acts as a terminator to detach the RNA polymerase from transcribing only, only and only when it is having poly stem loop immediately followed by UU. Not like this stem loop structure, and after few nucleotides UU, it will not act as a row independent terminator. Only stem loop stem. After the stem, immediately poly U should be there. Then only it will act as a row independent terminator. If it is something stem loop and followed by few nucleotides and poly U, it is not going to be row independent terminator. That is what the structure forms now when tryptophan is low. When tryptophan is low, ribosomes are going to stall uh, going to stop at the tryptophan codons because tryptophans are low. When it is stalled over there, so secondary structure will be automatically changed because it is disturbing the first secondary structure. So it will form something like this, and there is no intro independent terminator. RNA polymerase, which is at present somewhere here, is not going to stop. There is no terminator signals. We keep on transcribing. So it will not fall off. It will keep going the transcription, continue the transcription until the A. So then it will produce a long mRNA. And of course, as I mentioned to you earlier, so it will be having 5 ORFs, and it will produce 5 different proteins which are needed for the synthesizing the tryptophan molecules or amino acids which are needed when tryptophan is low. So that is the another the regulation of the tryptophan operon chosen by the bacteria uh, to effectively synthesize tryptophans whenever it is really needed, and don't want to synthesize the enzymes which are needed a lot of energy for the transcription and translation of the enzymes needed for the tryptophan operon, tryptophan amino acid synthesis. So it's not required when the tryptophan molecules are already abundant in the cell. So this is about the, so this is about the tryptophan, uh, the second regulation. So have you understand all of you clears now?